Malaria. Malaria is an endemic as the World Health Organization reports 200 million cases annually. It is responsible for 450,000 deaths in 2016, and it mainly affects populations in sub-Saharan Africa, as 90% of cases were reported there alone. Malaria is caused by Plasmodium falciparum, which is carried by the female Anopheles mosquito. The parasite is injected into the human blood when the mosquito carrying the parasite takes a blood meal. The parasite life cycle consists of 3 stages. The 3 stages include pre-erythrocytic, erythrocytic, and sporogonic. Although drugs can be used to treat malaria, the highly evolving nature of the parasite allows it to develop resistance, making it cost inefficient to continue developing drugs. The process of malaria begins with the pre-erythrocytic stage, when the sporocytes are released into the blood. Once released, they infect the hepatocytes, where they replicate in large numbers. When they replicate, the hepatocytes burst, causing merocytes to be released into the blood, marking the beginning of the erythrocytic stage. The red blood cells become infected, they burst, and their function is destroyed. This causes the onset of symptoms of malaria including fever, chills, and profuse sweating to take place. The sporocyte is coated by a circumsporocyte protein, antigen, that contains unique epitopes recognized by the immune system. Our vaccine aims to arrest malaria at the pre-erythrocytic stage. The vaccine design involves isolating the gene encoding the circumsporocyte protein. We will then insert this gene into the plasmid DNA and transform the yeast with the plasmid. Lastly, we purify the DNA plasmids carrying the circumsporocyte protein gene. When the gene is provided in the body, the circumsporocyte protein will be made and the immune response will be elicited. The tissue-resident mast cells respond by producing TNF-alpha cytokines, increasing the inflammatory response. As a primary immune response, the macrophages ingest the circumsporocyte protein. They then present the antigen to T cells and B cells in draining lymph nodes. The CD4 helper T cells activate B cells. The B cells then produce effector memory cells and anti-sporocyte antibodies in response to the circumsporocyte protein epitopes. CD8 T cells are activated and produce a cytotoxic response to the CSP protein. Adjuvants. There will be 2 adjuvants being used. The first adjuvant is the TNF-alpha cytokine, causing an increase in inflammation. This increase in inflammation will enhance the cytotoxic response of the CD8 T cells. The second adjuvant is a cell surface protein. We will incorporate a gene for an immune cell-specific cell surface protein in our DNA plasmid. It will help in delivering the circumsporocyte antigen to the immune cells. Dosage. Our vaccine targets populations in sub-Saharan Africa. The vaccine is designed to be a booster vaccine because of the parasite's nature to develop resistance. The first vaccination will be administered at the age of 3 and then every 10 years after. The vaccination will also be administered to those who are traveling to Africa, where there is a high prevalence of malaria. By being vaccinated, when one is exposed to the sporocyte, it will be able to elicit the immune response. Pipeline vaccines. RTS is the only pipeline vaccine that has gotten to phase 3 of the clinical trial. It is done by inserting a live attenuated parasite into the human. Our model is more advantageous because we are using a gene encoding for the antigen, thereby minimizing the risk of introducing a live parasite, since a live parasite is capable of reverting to a virulent parasite form. With the use of bed nets, medication, mosquito sprays, and our vaccine, We hope to put an end to malaria.